Hi everyone, John Manuel here, Spatial Science and Technology Advisor with Singular Genomics. Thank you for joining us today. We're excited to share some interesting and impactful information with you about our platform. I'll be opening our presentation called Spatial Histopathology, Perspectives and Implications of High Throughput In Situ Multiomics. After a cover of our technology and platform, I'll be joined by some colleagues of mine who will go deeper into each of the two areas they're covering, which include analysis and also sample prep for the G4X assay. So internally at Singular Genomics, we really have this ability to go deep on all aspects of pathology and histology. We can speak to the technical details at all stages of the spatial workflows, from sample procurement in clinical or surgical suites, through specimen handling and fixation, to tissue sectioning, and finally providing information and guidance on microscopic review and analysis. As a team, we cover the entire spectrum. So Dr. Jude Senaratne will be presenting the G4X viewer review, and he also serves as our expert pathologist, providing invaluable insights into microscopic examination across multiple tissue types and pathologies, translating findings into clinically relevant information, and supporting our robust analysis. I provide support on various topics from tissue procurement, dissection methods, multiple aspects of surgical pathology lab workflows and the nuances involved, and also help to manage projects with our customers. Gina Benedetto, she will be presenting histology sample prep for the G4X assay, and she is one of our scientists who also leads our histology section. She's bringing years of expertise and guidance on some of the most challenging histology specimens for spatial profiling. So together, along with many scientific and technical staff on our team, we have you covered. So to begin, I'd like to highlight how our G4X platform, together with some remarkable innovations in histology, are driving new insights and advancing our understanding in ways that complement the field of pathology. I'll also touch on how we're bringing unprecedented scale to spatial experiments like never seen before in the field. So I'd like to touch on how the G4X platform is improving understanding and broadening our insights in ways that relate directly to the field of pathology. When we think about high-resolution maps, the G4X platform allows us to capture molecular detail within intact tissue architecture. For pathologists, this allows the option to layer high-resolution multiomic data directly onto the structures they are already interpreting on a daily basis, and turning H&E images into rich molecular maps. In the world of biomarker discovery, the G4X profiling can reveal new biomarkers and therapeutic targets by showing which genes and proteins are expressed, and importantly, where they're expressed within the tissue or the cells. This spatial context provides critical insights into disease mechanisms and can guide the development of more precise therapies. Pathologists are leading the way with spatial, and that's something we recognize. Pathologists have a powerful ability to recognize patterns in tissue, interpret architecture, and connect those patterns with disease processes. It's this context that transforms raw molecular signals into something more meaningful in clinical research. And we will see and hear a little bit more about this from our staff pathologist, Dr. Jude Senaratne, as he demonstrates the G4X viewer a little later on. And advancing breakthroughs in precision diagnostics and personalized medicine is the final point I want to cover. Ultimately, we expect the partnership of spatial biology, pathology, and oncology to bring personalized medicine closer to reality by enabling molecular insights with the potential to be clinically meaningful. So I'd like to take a moment and talk about TissueStamp, how it's redefining histology and bringing spatial to scale in ways we have never seen before. So this truly is purpose-built tooling. This tooling was designed specifically with histotechs in mind. It is not a retrofitted tool. Rather, it's one that integrates seamlessly into the workflow people are already familiar with. That means minimal training, minimal disruption, and an intuitive experience that feels natural because it matches how histology labs already operate. Another benefit is sectioning without damaging blocks. So rather than taking the approach in creating TMAs, having the pull cores out of blocks, making them unusable in future or creating a degree of damage. What it does is it allows you to place sections from thin 5-micron sections that are taken off the block in routine histology practices. Now, with this tooling, although it is a nice alternative to punching FFPE blocks to create a TMA, if you have FFPE TMAs already, they are compatible with our platform, and I'll talk about that in the next slide because that can really introduce a third level of scale. We have our spread instrument, we have the multiple tissue sections, and then the potential for TMA. Also, our approach utilizing a gel pad and a user-friendly ROI selection process helps to decrease common issues such as curled tissue edges, folds, or missing tissues and gaps that sometimes occur in standard FFPE TMAs. It's a forgiving process in that it allows ROI selection in a way where if you choose the wrong section, all you have to do is take another 5-micron slice and then start again from there, which is a very simple process and minimizes the impact of the tissue that's taken from the block. Also, we have two size options, 10 by 10 millimeter or 4.5 by 4.5 millimeter. So very quickly to explain this, and this is an image, so if it doesn't make quite clear sense this time, don't worry about it because we do have a video coming up which Gina will present. So basically you have a tool which punches through a 5-micron thick section of FFPE tissue with a gel underneath. We have a grid system which allows you to load that with multiple tubes, we'll say, of gel and the tissue. We put a device on top of that that pushes the gels and tissue down onto the slide and essentially stamps it onto the slide, holding it in place, and we have a device that comes on top of that and locks that into place with just the right amount of pressure to transfer that tissue. So what do you end up with? You have options, 2 flow cells, one with 4 rows of 8, where you have 32 samples that can be put through, or 2 rows, each with 5, and you can put 10 samples through at 10 by 10 millimeters on the right or 4.5 by 4.5 on the left. Again, this is scaling experiments in ways that have never been seen before in spatial. So one of the biggest things that sets apart our platform compared to other commercially available solutions is the unparalleled throughput. With our system, labs can process dramatically more samples in the same amount of time or even less time. And what that really means is scalability. Researchers can take on larger studies or handle larger workloads, allowing for more efficient utilization of their staffing resources, of their time, making it a great solution to support large clinical studies. And as demand continues to grow, this capability does help to bring a degree of future-proofing labs. Also, all the lanes in our flow cells are fluidically independent. And what this allows are multiple experiments to be run side by side by allowing different tissue panels to be run in each lane. That maximizes instrument utilization and gives users a lot of flexibility. In practical terms, labs can run different panels simultaneously without the risk of cross-contamination, while also avoiding bottlenecks that arise when multiple researchers may need to coordinate the use of a single slide area. Regarding 3D reconstruction, our method allows multiple sections up to 128 to be taken and used for true 3-dimensional reconstruction. This gives researchers a clearer view of tissue structure and architecture in a more streamlined and efficient process. These improvements don't just enable better science, but they also save time and cost, delivering meaningful ROI for the lab, as we know is becoming increasingly more important. So we've discussed the throughput in terms of multi-section placement with TissueStamp, combined with the instrument speed of the G4X platform. But let's look at one more thing. So circling back to what I mentioned earlier, while our process allows a nice alternative to FFPE TMA construction, it still supports FFPE TMAs that were previously made. So if you look at just one of our flow cells, right, the 4 by 8 configuration with 32 samples at 4.5 by 4.5 millimeters, we can fit 32 sections of that into one single flow cell. Running 4 flow cells at once, that brings it to 128 samples. Now, if we were to set 2 cores, similar to how you would see in a TMA, that doubles that amount to 256 samples per run. Typically, the way that cores are set up or TMAs are made, you won't have two with this type of spacing where we see more spacing above and below the two cores. It's more likely to be 4 cores or maybe even more depending on the diameter of the core, such as 4 cores in this case. If you do have 4 cores in one of these 4.5 by 4.5 areas, what that allows is a total number of 512 samples to be put through in a single run. And a single run requires 1 day. So that level of scale we have not seen before in spatial, and we're very excited to be opening that up for users. This level of throughput is unmatched and offers a powerful solution for advancing large clinical trials and studies. And beyond throughput, the ability to process multiple sections also opens the door to generating robust 3D reconstructions, adding even more value to the data that we can deliver with the G4X platform. So let's get a little bit deeper into 3D reconstruction. What we have here are multiomic results generated with 10 sections, and this approach allowed us to build a complete 3D understanding of the tissue microenvironment, providing deeper insight into cellular organization, interactions, and function. In this case, we present spatial data from 10 serial sections of a renal cell carcinoma specimen, which include both tumor tissue as well as adjacent normal kidney. Each section was 1 by 1 centimeter, 5 microns thick, FFPE, and processed all together on one G4X flow cell in a single run. The analysis encompassed 6.2 million cells and 483 million transcripts, followed by unsupervised clustering. In these images, we restricted the analysis to a defined region. We characterized both the tissue and the tumor microenvironment through automated annotation of cell types and the identification of 3D tissue niches. So this enabled us not only to view the tumor and tissue microenvironment as a whole, but also to resolve specific cells and structures right down to the exact location in both the normal renal parenchyma and the area of the tumor. So by reconstructing tissue in 3 dimensions, as we have here, researchers can visualize complex architecture and cell-to-cell interactions with powerful clarity, revealing biological insights that remain hidden when using only traditional 2D analysis. So at Singular Genomics, we have positioned our early efforts primarily on human clinical FFPE samples, focusing on strong performance across a variety of samples that are relevant for clinical research. Our testing has included some of the most challenging specimen types for spatial, and many of you may be familiar with these, such as skin. We've had great results with skin. We've had solid results with pancreas as well. Strong data has been generated despite all that enzymatic activity that we know can be a threat to RNA. Even bone marrow, we've had really good data on, typically or historically a very challenging specimen to work with in spatial studies. Now, we've had these type of results despite not having an FFPE RNA QC process in place at this time. The G4X continues to perform well on samples that have gone through standard surgical pathology workflows, and that includes specimens that have been exposed to reagents known to negatively impact RNA quality. Building on this foundation, we are also expanding into mouse tissue, and our early results show success across several sample types and have been very promising. So beyond this, we are broadening our testing capabilities to include additional tissue types, further demonstrating the flexibility and applicability of our technology. Lastly, I do want to mention that if you don't see your tissue of interest here, it certainly doesn't mean that it won't work on our platform. We are busy running experiments in-house with additional tissues, working with many users or running other sample types, and we're gathering that information and data, and this list will continue to grow. If you have samples of interest, do reach out if you don't see them on here, and we'd love to connect with you and talk over those samples with you as well as your experimental interests. So I'd like to talk about one more thing, that's analysis, viewing, and platform capability. When it comes to integrated single-cell analysis, The G4X system delivers an end-to-end pipeline for single-cell research by automatically performing steps such as base calling, transcript detection, clustering, and cell segmentation. Together, these capabilities bring reliable single-cell data outputs. When we think about the viewer of that multiomic data, users can seamlessly overlay fluorescent H&E images, tissue images with protein expression maps, and spatial transcript localization data. This allows them to achieve subcellular level resolution that reveals those intricate relationships between tissue morphology, protein distribution, and transcript positions. And lastly, the system is plug and play with third-party platforms. The system produces standardized spatial data outputs, and these outputs are compatible with your preferred open source and commercial secondary analysis tools. This allows for deeper insights, of course. It allows for flexibility, so you can integrate these results directly into your preferred analysis pipelines, enabling deeper exploration, more advanced interpretation, and the ability to tailor insights to your specific research objectives. So with that, I'd like to hand it off to Gina Benedetto, who will be presenting histology sample prep for the G4X assay.
Thank you, John, for that introduction, and thank you so much to everyone who's tuning in online. For this portion of the talk, I'm going to be covering the histology sample prep needed for the G4X spatial assay. My name is Gina Benedetto. I am a histologist and a spatial application scientist here at Singular Genomics. We have a quick agenda for today. I'm going to be going over 3 main portions of the assay. So the first is going to be the histology sample prep, and this is divided into 2 sections. It's going to be the tissue sectioning workflow specifically, and then from there I'm going to cover the gel tray mounting workflow. And then lastly, I'm going to be covering an overview of the TissuStamp workflow. And this is our tissue transfer workflow where we get those tissues from the intermediate gel pad trays onto your glass slides. At the very bottom here, this is just a very high-level end-to-end overview of the G4X workflow. And so this is just further illustrating that I will just be covering the upstream portions of the workflow from tissue sectioning to tissue transfer. Before I dive into the technicals, let me give you some context. So why TissuStamp? Singular Genomics saw a need in the sense that spatial biology assays often require these highly specialized sample preparation techniques. And these techniques can be challenging for histologists that have been in the field for many years. Specifically, I'm talking about block scoring, which, other than the G4X assay, is the only method to isolate small ROIs from whole FFPE tissue block faces. And the second is this single attempt precision slide mounting that can be very unforgiving. Spatial assays are also very limited in the surface area that's available for analysis. So often you're working in this very small area on a standard size glass slide, and this is going to severely limit your throughput capabilities. So the purpose of TissueStamp is to reduce or completely eliminate these high complexities that I just touched on the previous slide. So TissueStamp is our novel tissue transfer system. What that's going to do is it's going to maximize the available area of a standard glass slide so you can fit as many samples onto that slide as possible, which is going to boost your throughput. And it's also going to completely eliminate these high-risk sample preparation procedures that I talked about, namely block scoring and the single attempt slide mounting. And that's what's going to broaden accessibility across a wide range of technical skill levels for your histologists and other lab users. Okay, with that, let's dive into the first part of the workflow that I'll cover, which is the sectioning workflow for the G4X assay. And this is going to be what eliminates the need for block scoring. What is block scoring? So when you have assays that are really rigid in their available surface area for analysis, you have to find a way to isolate a smaller ROI from a large tissue piece. Besides the G4X TissueStamp system, the only way to do this is to score the block using a razor blade. This is not a very refined system, so there's no way to standardize how deep you go into the block with this razor blade, essentially how many microns into the block you're going, because you're just doing it manually. And also, once you score a block, there's no going back. The only thing that you can do to achieve a full face again is to shave through entirely all of that scored area. So over here you can see what a ribbon looks like when you cut it from a scored block. And for other assays, what you would need to do is isolate one of these very small pieces within this tissue section and float and mount that onto a glass slide, which is a very complicated procedure. Over here, you can see what a standard ribbon looks like from a block face that has not been scored. And this is the kind of ribbon that you would cut for the G4X assay. Okay, now that the sectioning workflow is done, we're going to move on to the gel tray mounting workflow. This is the workflow that's going to eliminate that unforgiving single attempt precision slide mounting that's common to other spatial assays. So these are my hands actually. You can see I've just floated 3 whole FFPE tissue sections onto this tissue collection bath. This workflow mimics a standard histology section collection procedure. So I'm doing the same thing I would otherwise, except instead of mounting on a glass slide, I'm mounting onto this gel pad tray. The benefit of the gel pad tray is that if I'm unsatisfied with the location or the placement of these sections, or if there are minor imperfections like wrinkles or ripples in the tissue, what I can do is I can resubmerge this gel pad tray into the water bath. I can release my tissue sections, and then I can remount them onto that gel pad tray pretty much until I'm satisfied. And the other thing is that this gel pad tray, it mimics the size and shape of a glass slide. So you have this handle here that's easy to hold, and it's also a great surface area to label your specimens. Once your tissue is mounted onto this tray, it can be stored for up to a month at 4C. Okay, now we're going to dive into the TissueStamp workflow. So once my tissue sections are mounted onto the gel tray, they're ready to be punched out to isolate my ROIs. So we have 2 formats available for these blades. One is the 4.5 by 4.5, and the other one is a 10 by 10. And so I'm going to start by inserting this blade into this handle here, and that will form this assembled punching tool. So this mechanism is very similar to a biopsy punch, and what you'll do is you'll isolate your desired ROI from each tissue section, and you will simply punch it out of the gel. This is just further illustrating how these formats work in terms of ROI selection. So instead of taking a razor blade and trying to carve out these ROIs, what I can do is I can plan where I'm going to stamp out the ROIs using a simple scaled photo of my FFPE block face. So here's the small format on the left and the large format on the right. And so I can take multiple punches from one tissue section, or I can take most, if not all, of the tissue section from the large format. And what I can do is, using the corresponding H&E, I can sort of map out these precision ROIs. So this is a tonsil section. You can see here that with these punches, I've tried my best to maximize the lymphoid tissue while also minimizing this sort of peripheral tissue over here, like this is skin, for example, and trying to minimize these connective tissue layers so I can isolate my germinal centers with my immune cell expression. Moving on to the second half of the TissueStamp workflow. So this is what's going to reduce these restrictive tissue size limitations, and we're going to maximize the available space for tissue on a standard size glass slide. So after I have punched my ROI out of a tissue section, I'm going to remove the blade, I'm going to invert it, and then I'm going to place it in this black template that you see down below here. Underneath that template is a glass slide mounted face up. The glass slide is a standard size. but it's worth mentioning that it's unique in the sense that it's coated in a proprietary adhesive matrix, and this matrix is designed to have strong sample retention across a bunch of different tissue types like colon, kidney, lung, and tonsil, and it even shows strong sample retention across some particularly difficult tissue types that we come across, like skin, bone marrow, or some other adipose-rich tissues like breast, for example. And so once you've loaded as many punches as you desire into this template, you can assemble this pusher device on top of those punches. And so each of these pistons is individually spring-loaded, so you can fully load this template if you would like, but if you don't want to, nothing is going to be off balance. And each of your gel punches and the tissue mounted underneath are going to get the same amount of pressure, so everything is standardized. Once this pusher device is assembled on top, you can take this handle, gently lift it over, and it has this soft snapping mechanism to it, so that once you've assembled all the pieces together, nothing's moving around and stretching the tissue out, and everything is secure. Once you've mounted your tissue onto glass slides, you can store them for up to 4 to 6 months. In this slide, I'm further illustrating how we are really maximizing the available area of a standard glass slide with tissues. So here we have our two formats, the 4.5 by 4.5 on the left and the 10 by 10 on the right-hand side. And so for the small format, you can have up to 32 individual samples if you wish. And for the larger format, you can do up to 10. And so this is using all of the surface area that we possibly can on a glass slide. So this is really going to boost your throughput capabilities. Also, each one of these lanes is fluidically isolated, so you can run up to 4 or up to 2 different experimental conditions on these slides, and that includes different tissue-specific panels and anything like that, and you're not going to have any cross-contamination or leakage between those lanes. So I've included a lot of photos for you today to illustrate the workflow, but in case you are a highly visual learner like I am, we also do have these instructional videos to supplement. So there's one for sectioning and one for tissue transfer. I'll give you a second now to take out your phones and scan these QR codes so you can have access to these videos. And that way you can see everything flowing together and someone doing it in real time with narration, just so you can put all of these steps together and more easily conceptualize the entire workflow. So that concludes my portion of the talk where I cover just the upstream histology sample prep required for the G4X assay. Thank you again to everyone who's tuning in today. We really appreciate it. With that, I'm going to hand things off to Jude, our pathologist. He's going to walk you through the visualization of resulting tissue sections in our G4X viewer that result from a G4X Multiomics run. Thank you.
Hello, everyone. Thank you for joining the call today, and I'm going to do a G4X viewer. My name is Jude Senaratne. I'm working for Singular Genomics as the Senior Staff Pathology Scientist. When you open the G4X viewer, you have the option to select multi-file or single file mode. I like to use the multi-file mode as I'm going to upload 4 different files today. The viewer, we are keep on making some improvements. The current version you are looking at is 2.6.2. The client will receive 6 different files, which they can upload to the G4X viewer. Out of those 6, today I'm going to use 4 of those files. For the remaining 2, I'm not going to use. One is metadata JSON file, and also another file called nuclear OMET file. Nuclear details or the nuclear stain already included in the protein panel. Therefore, I'm not going to use those 2 files. Let me go ahead and upload the remaining 4 files. One is the OMET file, which have all the protein data. That's the first one I'm going to upload. Now you can see different protein markers being uploaded. I go to the protein channel settings, and there I can change the different markers. The first one I would like to add is the nuclear marker. Here in these three dots, I can change 8 different colors. I like blue. Sometimes I use white. Today I'll use blue. And upper and lower intensities right here being selected by a automated algorithm. You can adjust them depend on how you want to visualize your analyte of interest. Second channel, let me go ahead and turn on pan-CK. Now you can see the tissue anatomy a little bit favorably. I would like to have ASMA as well, and also to visualize blood vessel endothelium CD31. Here, the blood vessel endothelium, I would like to set it up, change the color to orange. Now you can see this is the mucosa, this is the submucosa and muscularis layer. What we are looking at is a colon. It is not a healthy colon because it has some cancer spots, correct? 1, 2, 3, 4. 4 spots being highlighted with pan-CK, indicating that there is invasive carcinoma. In this middle muscular layer, that's called muscularis mucosa. Anything above the muscularis, anything towards the mucosal side, that's non-invasive. Anything below this one called invasive adenocarcinoma. So what we are looking at is a invasive adenocarcinoma. Let me upload the other file, the H&E file. So I go to the bright field image settings. Here, I'm going to add, drag and drop H&E OMET file. You can do it by left clicking on this button as well. To turn on, I'm going to click on this circle. Now you can see the epithelium highlighted nicely and the tumor area, or this is more similar to a classic H&E. And this is the tumor area, tumor cells, and there is some a nerve, and also there are some lot of immune cells or inflammatory cells, correct? So let me go ahead and turn off these channels. Let me add the transcripts file. I'm going to add the .tar file in the transcript file location. It will take some little time. There you go. You can see the transcripts. Let me go to the channel controls for proteins and turn off the other channels, just leaving the nuclear channel only. And then I go to the transcript layer and going to change different settings. Right now, only visualize 0.16% of the transcripts that's available. So you can visualize individual transcripts as well. Let me turn on CEA CAMP5, which is specific for colonic adenocarcinomas and the normal colonic epithelium. Let me increase this percentage to maybe 20%. Now you can see the normal epithelium being highlighted, and also it's well picked up the tumors. Likewise, you can study the relationship between the transcripts and the proteins. Like, for example, you can turn on immune cells, you can turn on pan-CK. Pan-CK should highlight on top of this CAM5. And if we turn on, let me add another channel, another protein channel, and CD8 cytotoxic T cells. Let me change the color to white. And now these cells, a little bit farther away from these signals, correct transcript signals. So you can study the relationship between transcripts and proteins in this image. So you can explore more biology. I'll go to the view settings and turn off transcript layer. And then let me upload the fourth file we have, the bin file, to see the cell markups. I'm going to add it to the cell mask right there, and it will take some time to upload. Now the cell mask is uploaded here. If I go down and go to the segmentation layer settings, here, if the markups are not clear, you can increase the brightness. Let's put it to 68%. Now it's clear. The cell markup have different colors because we use transcript layer details to categorize or cluster similar cells. Therefore, you can see these epithelial cells being clustered in green color, these muscle cells, smooth muscle cells clustered in brown color, and there are some other cells in different colors, which we can explore more. And these cancer cells even are different than these epithelial cells because cancer cells, it been highlighted with a different marker, different transcripts that express than the normal epithelium. You can filter these ones, you can turn off and you can turn on different markers, and you can see the markups without any colors, just pure white, and you can go to a higher magnification, and by turn on and off the nuclear markers, you can see whether this classification did a good job or not. Okay, so because now I'm making this activity, I would like to turn off pan CK, and for the nuclear channel, I would like to go with the green color. And I'm going to change the lower intensity to 100 and the upper intensity maybe to another brighter by reducing them to 800. Now I can see clearly nucleus. Let's see how the segmentation is doing for us. Now I'm going to turn on and off. So far the segmentation looks pretty good. There are here and there are a few misses, but overall the segmentation is happy. If you're not happy with this segmentation, you can do your own segmentation as well. I think this concludes my talk today about G4X Viewer. If you have any questions, please email me and I'm happy to answer any of the questions that you have. Thank you.